Hello everyone, let's get started. In today's lab supplement video, we're going to be discussing more metabolic tests. Be sure to take a good set of notes for yourself while you watch the video and keep them in your laboratory notebook. Today's metabolic tests will include the Nitrate Reduction Test, the Motility Indole Ornithine or MIO test, and the gelatin hydrolysis test. Before we begin discussing the particulars of these metabolic tests, we need to do a little review of basic chemistry. Many of the metabolic reactions that occur inside of living microbes or living cells in general are called oxidation-reduction reactions. Remember, in chemistry, the word oxidation refers to a loss of electrons. The word reduction refers to the gain of electrons. During these oxidation-reduction reactions, if one molecule gets oxidized, if it loses electrons, then another molecule must be reduced. It has to gain electrons. Those two processes, in other words, always go together. Oxidation cannot occur without reduction, and vice versa. The loss of electrons occurs during catabolic reactions, when electrons are released because covalent bonds are being broken down. The gain of electrons occurs during anabolic reactions, when new molecules are being built, new covalent bonds are being built between atoms so that they are sharing a pair of electrons. Remember, too, that all microbes, in fact, all living cells, use a molecule called adenosine triphosphate, or ATP, as their energy currency. ATP is used to supply energy to cells. And the way it gets used is by having its outer phosphate group removed and then put back on, and then removed and then put back on over and over and over again. Every time the phosphate group on the outside of ATP gets removed, the covalent bond holding it on gets broken. And when bonds are broken, electrons get released from that bond. And electrons are the carriers or the conduits of energy. So when ATP is broken, energy gets released. When that phosphate group gets put back onto ATP, a new covalent bond is built. Electrons get stored in that bond, and that process, that anabolic process, requires energy, consumes energy. So ATP gets broken apart, and a little energy gets released. ATP gets rebuilt, and a little energy gets consumed. That's how ATP gets used in a cell. ATP gets made in a cell very differently. ATP gets made during a process called cellular respiration. In eukaryotic microbes, cellular respiration occurs in an organelle called the mitochondria. In prokaryotic microbes, there are no mitochondria. There are no organelles in bacteria and archaea. So when they make their ATP, they do it at the plasma membrane instead of the mitochondria. Cellular respiration is an incredibly complex process. It involves a long series of reactions that we can divide into 4 separate processes. It begins with glycolysis. It moves into pyruvate processing, followed by the Krebs cycle, and finally electron transport. All cells use carbohydrate for cellular respiration, specifically glucose. It's glucose that will be entered into this process of glycolysis. It's glucose that will be slowly broken down, one bond after another, until all of the electrons have been released from its covalent bonds. Glucose, in other words, gets oxidized during cellular respiration. Cellular respiration is going to take glucose, which is a 6-carbon molecule, all the way down to carbon dioxide. Cellular respiration is going to break all of the covalent bonds in glucose, one by one, releasing all of those electrons until the only thing that's left is carbon dioxide. Glucose gets oxidized. It is losing electrons during cellular respiration. The electrons that get released from glucose are going to be fed into the electron transport chain during that last set of reactions called electron transport. As the electrons get passed down this structure called the electron transport chain, their energy is going to be harvested. The energy that those electrons are carrying with them is going to be harvested. Now, at the end of the electron transport chain, the electrons need to go somewhere. They have to be received by a final electron acceptor molecule. In other words, something has to get reduced. And that something is this final electron acceptor molecule. Now, in aerobic microbes and in other aerobes like humans, the final electron acceptor molecule at the end of the electron transport chain is oxygen gas. In other microbes, though, cellular respiration doesn't use oxygen gas as the final electron acceptor. Instead, those microbes perform cellular respiration anaerobically. They use something other than oxygen as their final electron acceptor. Now, there are many molecules that can be used for this purpose, but the most common ones are nitrate, sulfate, and carbonate, with nitrate being most common. Now, just for completeness' sake, remember we said the electrons get passed down the electron transport chain and their energy gets harvested from them. Remember that after cellular respiration, there's another set of reactions that occurs, and those are called oxidative phosphorylation. During oxidative phosphorylation, the energy that was harvested from those electrons, the electrons that came out of glucose, that energy is going to be used to make ATP. So it's important to remember that all microbes, all cells actually, use carbohydrate as the primary substrate for ATP production. It doesn't matter if they're performing cellular respiration aerobically or anaerobically. Glucose is going to be the molecule that goes in to cellular respiration. Glucose gets oxidized. Remember, that means broken apart, loss of electrons. It gets oxidized all the way down to carbon dioxide during cellular respiration. All of the electrons that got lost from glucose, that got released from glucose, will be fed down the electron transport chain so their energy can be harvested and used to make ATP. The final electron acceptor molecule sits at the end of the electron transport chain. That's the molecule that's going to receive the electrons that were released from glucose. That's the molecule that's going to get reduced. Glucose gets oxidized, the final electron acceptor gets reduced, and that completes cellular respiration. That brings us to the first metabolic test we're going to discuss today, which is the Nitrate Reduction Test. The Nitrate Reduction Test is used to determine two things. Number one, if a microbe uses nitrate as its final electron acceptor, and two, how far that microbe can reduce the nitrate molecule. Let's take a look at some equations. In aerobic organisms like you and I, oxygen gas is the final electron acceptor at the end of the electron transport chain. Oxygen is going to get reduced with all of the electrons that came out of glucose. Glucose gets oxidized, the final electron acceptor gets reduced. Let's look at the equation for that reduction. The oxygen gas is going to take on the electrons, that's the E minus here, as well as some hydrogen ion, and water will be produced. Now, note that this equation is not balanced. We don't need to balance this equation to understand it. Oxygen is going to gain electrons. It's going to get reduced. And with the addition of a little hydrogen ion, water gets produced. What about the anaerobes? What about the microbes that don't use oxygen during cellular respiration? Well, most of them are going to use some other inorganic molecule, some molecule that has no carbon in it, but does have oxygen in it, something like nitrate or sulfate or carbonate. One of these molecules is going to be at the end of the electron transport chain, and it's going to receive the electrons. Nitrate, as I said, can function as one of these final electron acceptors. And the interesting thing about nitrate is that it can be reduced in stages. Nitrate, which is NO3 minus, can gain electrons and produce a molecule called nitrite. That's NO2 minus, as well as a little bit of water. So nitrate gets reduced to nitrite. But here's the thing, nitrite can be reduced further. Nitrite can also gain electrons and produce a molecule called nitrous oxide. That's a reduction reaction too. And we're not done yet, because nitrous oxide can also be reduced. It can gain electrons and produce nitrogen gas. Now we're done. Nitrogen gas cannot be reduced any further. It turns out that some microbes that use nitrate as their final electron acceptor are going to follow this first pathway and then stop. They're going to reduce nitrate to nitrite and then stop. But other microbes will continue to perform this reduction. The nitrite will be reduced to nitrous oxide, and then they'll stop. In still other microbes, the reduction continues, and the nitrous oxide gets reduced to nitrogen gas. So microbes can perform all different combinations of reduction pathways for nitrate, depending on the species. And that's what the Nitrate Reduction Test is looking at. Now, just for completeness sake, remember that some microbes use sulfate as their final electron acceptor. Sulfate can be reduced to produce hydrogen sulfide and water. Some microbes use carbonate. When that molecule gets reduced, we end up with methane and a little bit of water. But we're going to focus in on the pathways of nitrate reduction with this test. So, the nitrate reduction test is primarily testing for the ability of a microbe to reduce nitrate to nitrite. That's the first part of the test. Nitrate reduction uses a broth, and that broth has been supplemented with potassium nitrate. What we do is we inoculate the nitrate reduction broth and we incubate it for a full 48 hours. The reason we incubate it so long is because we want to give the microbe enough time to follow through as far as it can with the reduction of nitrate. When we take the nitrate reduction tube out of the incubator, we're not going to read it right away. Instead, we're going to add some reagents to it and then read the result. We're going to add 2 reagents to the tube. We typically refer to them as reagent A and reagent B. These are actual chemical compounds, but for ease of speech, we refer to them as A and B. We add those reagents into the tube and we look for a red color change. If the tube turns red with the addition of these reagents, it means that the nitrate in the broth was reduced to nitrite. Now, sometimes we put in reagent A and B and there is no color change. We're not done yet. At that point, after we add A and B, if there's no red color in the tube, we're going to add another reagent. We're going to add a zinc reagent. It's actually zinc powder. And what that zinc reagent will do is force the reduction of any nitrate that's in the tube. The zinc powder will force nitrate to nitrite, and the tube will turn red. If the bacteria, in other words, didn't reduce the nitrate, we'll use the zinc powder to do it. That will tell us that the nitrate was still in the tube. When we add the zinc powder, if the tube turns red, it's because the zinc reduced nitrate to nitrite. Now, after we add zinc powder, sometimes there is still no red color. What do we do then? Well, at that point, we know that the bacteria must have reduced the nitrate beyond nitrite. It must have gone to either nitrous oxide or nitrogen gas. The question is, how far did it go? Which product was produced? Inside the nitrate reduction broth, there's a tiny little test tube that is placed upside down into the broth. This is referred to as a Durham tube. The reason we place Durham tubes like this is because they will catch any gas that gets produced during a metabolic reaction. In this case, if the nitrate gets reduced all the way to nitrogen gas, that nitrogen gas will collect as a bubble inside the upside down Durham tube. If there is no gas in the Durham tube, we know that the final product was nitrous oxide. Now, the nitrate reduction test is very complex, and it's hard to wrap your head around all of the steps and what each step means. So let's summarize it once again. We're going to take a tube of nitrate reduction broth and we're going to inoculate it with bacteria, then put it into the incubator for 48 hours. When we take the tube out of the incubator, we're going to immediately add reagents A and B. If the bacteria that's growing in the tube took the nitrate in the broth and reduced it to nitrite, once we add reagents A and B, they're going to combine with the nitrite to produce a red color. That's a positive test result. That tells us that the bacterium reduced nitrate to nitrite. the nitrite reacted with reagents A and B and turned the broth red. If there's no red color after A and B are added, there's two possible explanations for that. Number one, either the nitrate is still in the tube, in other words, there was no reduction, or the bacterium did reduce the nitrate, but it didn't stop at nitrite. It further reduced the nitrite, either to nitrous oxide or all the way to nitrogen gas. Two possible explanations, but which one is correct? How do we know? The next step, if the tube does not turn red after the addition of reagents A and B, is to add the zinc reagent, zinc powder. If nitrate is still in that tube, remember that was explanation number one, the zinc reagent will reduce the nitrate to nitrite. And the nitrite will then react with the A and B that's in there, and a red color will be produced. But this is a negative test result, because in this case, the microbe didn't reduce the nitrate, the zinc powder did. The nitrate was still in the tube. The bacterium wasn't able to reduce it. So we added in the zinc powder, and the zinc reduced the nitrate to nitrite, turning the tube red. That is a negative test result. But what if there is no color change after we add the zinc reagent? Well, that means the second explanation must be correct. The bacterium must have reduced nitrate to nitrite and then further. So in that tube, there's either nitrous oxide, which dissolves into the medium, or nitrogen gas, which will collect in the Durham tube. Either way, the nitrate got reduced, so that is a positive test. How do we know which product was left behind? Well, we look for gas in the Durham tube. If there's gas, if there's a bubble of gas trapped in that upside down Durham tube, it's nitrogen gas. If there is no gas, the final product was nitrous oxide. Now, in your lab handout, you're going to find this flow chart to help you work through your interpretation of the nitrate reduction test. Again, the broth gets inoculated, incubated for 48 hours. When it comes out of the incubator, we add two reagents, A and B. If the broth turns red, we've got a positive result for nitrate reduction. If the tube remains colorless, we have more work to do. We add the zinc reagent. If it turns red, that's a negative test result. That means that the bacterium was not able to reduce the nitrate. The zinc dust reduced the nitrate. If the tube is still colorless after the zinc dust gets added, we have to look at the Durham tube. If there's gas present, we know that the reduction of nitrate went all the way to nitrogen gas. Positive test result. If there's no gas in the Durham tube, we know that the nitrate was reduced to nitrous oxide. Positive test result. In this image on the right side, you can see what nitrate reduction broth looks like before it gets inoculated. This is what we mean by colorless. And this is that red color that we look for. The second test that we're going to be performing in the lab is the motility indole ornithine, or MIO test. This test is actually a set of 3 different tests. This picture right here shows us MIO medium. It's actually a semi-solid medium. It feels a lot like agar. It contains an indicator dye that is going to change color as pH changes. We've talked about that before. This test is going to test for whether or not the bacteria is motile, whether or not it can produce a molecule called indole, and whether it can produce an enzyme called ornithine carboxylase. So 3 tests in one. The way we're going to test for motility is by how we inoculate the tube. We're not going to use our typical inoculating loop, but instead we're going to use an inoculating wire. We're going to place cells on the sterile inoculating wire, and we're going to stab the wire down into this agar. That's going to leave cells behind in this narrow tract created by the wire. Motile organisms can leave that tract as they grow. They can actually swim out of that stab line and into the rest of the tube. If the organism is not motile, it will only grow within the stab line. For the production of indole, we actually have to use a reagent after we take the tube out of the incubator. Indole is a product that's produced when an amino acid called tryptophan gets broken down. This is called a deamination reaction. And all that means is that the amino group from the amino acid tryptophan is going to be removed, producing a molecule, again, called indole. We're going to be able to tell if indole is present when we add a reagent called Kovacs reagent. The Kovacs is going to react with any indole and produce a red color. We're going to see this red as a ring at the very top of the tube. If there is no indole present, the Kovacs reagent is going to remain a yellow color. The final test is for the enzyme called ornithine carboxylase. Ornithine carboxylase converts a different amino acid called ornithine to a molecule called putrescine. That's a great name, right? This is not a deamination reaction. This is a decarboxylation reaction. During this reaction, the carboxyl group is removed from the amino acid ornithine, producing this molecule called putrescine. Now, putrescine is acidic. It's going to lower the pH of the medium, and it's going to turn a faded yellow color. So from this purple color, we'll see a faded yellow develop if putrescine is present. Now, these 3 tests together are going to help us differentiate gram-negative enteric bacilli one from another. Many of the organisms that live in the gastrointestinal tract are gram-negative rods, and they can be hard to differentiate one from another. In particular, the MIO test helps us tell members of the Enterobacteriaceae family apart. E. coli belongs to this family, as does another gram-negative rod called Enterobacter aerogenes. So this is a test that will help us tell these 2 organisms apart, as well as lots of other gram-negative enteric bacilli. On this slide, we're looking at some of the results that we can get from an MIO test. On the upper left, we can see the difference between a positive and a negative result for motility. Now, this medium that you're looking at, this is not MIO medium. This is medium from a different test, but it also happens to test for motility. And because the medium is transparent, it's a lot easier to see the stab line than it is in an MIO tube. Over here you can see a line right in the middle of the agar. It looks a little bit cloudy in there, and that's because bacteria are growing in there. The bacteria were stabbed down into the agar with the inoculating wire. They grew, but they were not able to swim into the agar. They stayed right in that stab line. Over here you see something different. First of all, the stab line is harder to make out over here. You can see it down here, but up here it just looks like a big cloudy area. The bacteria that were stabbed down into the agar are motile bacteria, and as they grew, they were able to swim away from the stab line into the rest of the agar, producing this sort of a cloudy, widened look to the stab line. Motility negative, motility positive. Down here, we're looking at what a positive reaction looks like for the production of indole. Now again, this is a different test medium. This is not MIO, but we're going to see something very similar for an MIO test, and that's right at the top of the tube. Remember, this is agar. When it comes out of the incubator, we're going to add a liquid reagent called Kovacs reagent, and it will sit right at the top of the tube. If indole is present, it's going to show a red color. If there is no indole present, the Kovacs will remain sort of a dull yellow color. So we're looking at the ring that forms at the top of the tube to determine whether or not indole has been produced. The final test is for ornithine carboxylase. You're looking at 2 tubes of MIO medium here. Remember, it starts as a purple color. Over here, I just want you to notice that we've got a motile organism. The stab line looks very wide, and that's because the cells that were placed in here started to swim out of the stab line into the agar. At the top of the tube, you can see the Kovacs reagent, but I want you to notice that the medium itself has turned yellow. This means that putrescine is present. It has reduced the pH of the media, and it has caused it to change color to this sort of pale, dull yellow. Putrescine is produced if the bacterium has the enzyme ornithine carboxylase, and it can convert ornithine to putrescine. So for this tube, we would say we have a positive result for motility, we have a negative result for indole, and we have a positive result for ornithine carboxylase. Over here, you can't see the stab line because of the way the picture was taken, so we can't evaluate motility. But over here, we also have a negative reaction for indole, and we have a negative reaction for ornithine carboxylase. The final test that we're going to discuss today is the gelatin hydrolysis test. This test uses gelatin medium, which has a semi-solid to solid consistency, just like agar. Gelatin is just a protein, and it's derived from collagen from animal tissues. The gelatin hydrolysis test is going to test to see if a bacterium produces an enzyme called gelatinase. Gelatinase is an enzyme that actually gets secreted by certain types of bacteria. It gets secreted out of the cell and into the environment. If there's any gelatin in the environment, the gelatinase will break it apart into its component amino acids. Remember, all protein is made out of a chain of amino acids. The gelatinase enzyme is going to help the bacterium break down the gelatin into amino acids, so those amino acids can then be absorbed by the bacterial cell and used to make more protein. When this reaction occurs, the gelatin medium is going to go from this semi-solid consistency into a liquid. There will be no gelatin left in the tube, just amino acids. And without any gelatin in there, there's nothing to solidify the medium. Now, one of the interesting things about the gelatin hydrolysis test is how we incubate it. We have to incubate it at room temperature, and we have to leave it to incubate for anywhere from 5 to 7 days. It takes that long for enough cells to grow for us to determine whether or not the gelatinase enzyme is present. Now, why don't we just put it into an incubator? The answer is that heat in the incubator will also cause gelatin to liquefy, so we can't do that. If we took a gelatin medium tube and we put it into an ordinary 37-degree incubator, the heat in the incubator will liquefy the gelatin. It's not going to tell us whether or not any gelatinase was made, because the heat is what would turn it to liquid. So unlike agar, gelatin doesn't stay solid at 37 degrees Celsius. For that reason, we have to incubate at room temperature. This gelatin hydrolysis test is going to help us to identify different types of bacteria that are known to be gelatinase positive. Staphylococcus species, for example, members of that Enterobacteriaceae family that we mentioned earlier, they are going to be positive for the production of gelatinase. And some of the gram-positive bacilli will also produce a positive result.